Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
So the next speaker is Lisan Wang from University of Pennsylvania, and he will be talking about scaling genome rearrangement phylogeny reconstruction.
Thank you. First, thank you for having me here. And again, I'm going to talk about something that's a little bit different, genome rearrangement phylogeny. And in particular, I'll talk about what the genome rearrangement phylogeny problem is and what are the difficulties for solving large data sets for genome rearrangements and how we try to overcome the difficulties. So there are different kinds of genomes that one can look at. In particular, we're focusing on plant chloroplasts. Here is the chloroplast genome for the tobacco. And you can see it's a circular chromosome, and the locations as well as the which of the two strands of each gene is labeled here. So a chloroplast, plant chloroplast genome has about 120 genes, and the number is quite constant across different plant species. And there's another thing that's really good here. So as you can see, there are two inverted repeats here. They're symmetric around the replication origin. They have the same sequence, so they have the same gene orders, but in opposite directions. But other than that, there are no duplicates in the genes. So generally speaking, the genomes across different species for chloroplasts, they have roughly the same set of genes, no duplicates, and we can identify the locations with no ambiguity, generally speaking. So that makes this a really nice model for studying the mathematics of genome arrangements. So to formalize the problem, we can abstract a genome like this. We have two concentric circles corresponding to the two DNA strands, and the arrows represent the locations of genes, and here they are labeled. And you can start from any gene and go in either direction and write down the orders of the gene labels, and you can put signs in front of them to indicate which of the two strands they're on. So you can start from gene 1, and then the next one you encounter is gene 5, but on opposite strand of gene 1, so you put a minus sign in front of it, and so on. So you can have this representation as well as this representation starting from 5 going clockwise. They give you, they correspond to the same circular gene orders. So the problem of genome rearrangement phylogeny is this. Given the input gene orders, in this case they're circular gene orders, you want to find a tree where the leaves correspond to these gene orders such that this tree explains your data the best. For example, they have the lowest Parsimony score, highest likelihood score, or so on. So notice that here this tree is unrooted. Of course, we know that there is a direction, well, in reality, there's a time direction. So the reason why it's unrooted is the same case as a DNA protein phylogeny. If you look at the mathematical definition of the models that people are studying for evolution, they are actually, these models are time reversible, meaning that you can take a tree topology and root it at any different location. They give you exactly the same score. So what we want to find is this unrooted tree topology. And here are some rearrangement events that people look at, biologists look at. Starting from this genome with 10 genes, we can take a segment, break it up, but place it at the same place. At the same time, the order is inverted. So this event, we call it an inversion. So when you do this inversion, all genes jump to opposite strands. So you change their signs. So the order and signs are changed. A transposition is an event that moves the segment to a different location without changing its direction or the order of genes. And inverted transposition is a transposition followed by an inversion on the transposed segment. So the first thing is, if you look at these events, they change the order, they change the sign, but they don't change the gene content. You don't have duplicate genes, you don't have deleted genes. The gene content is conserved. And the second is, if you look at inversions, for example, all possible inversions, they're sufficient to transform any gene order to any other gene order. So, and the same thing with transposition, inverted transposition as well. And these different events, actually in reality, they happen in different scenarios. For example, biologists believe that in plant chloroplast evolution, you can explain all the things just using inversions, whereas for animal mitochondria, it's mainly transposition and inverted transpositions. So they actually correspond to different scenarios. Well, why do we want to study this? Well, it's an interesting problem algorithmically, mathematically, and these events happen, these are rare events. They don't happen like every day or every year. They're rare changes, and they have a much larger state space. Whereas in DNA sequence, it's a sequence with every site has 4 different states, protein 20 different states. A gene order is really a single character with the number of states being the number of different permutations together with signs. So it's a huge state space. So what that implies is, well, you're going to expect the evolutionary process have lower homoplasy. Homoplasy meaning that you start from one state, going to another state by mutation, then going back to the original state. So in that case, you couldn't observe the event. For DNA, that's really easy. You only have 4 states. But for gene order, that means you have to have exactly the same inversion to go back, which is a lot less likely. So because of these things, we hope that studying these, this evolutionary process allows us to look at deeper evolutionary events that happened a long time ago because of the higher signal-to-noise ratio. And of course, it's a different evolutionary dynamic process, and we can we want to look at how what is the evolutionary implication in, say, the organization of genes in the chromosomes, and so on. So the first question for studying gene rearrangement is, of course, given any 2 gene orders, we need to be able to compare them. There are different kinds of edit distances one can look at. The first one is the celebrated inversion distance, which is you take any 2 gene orders with the same set of genes with no duplicates. What is the minimum number of inversions required to transform one gene order into the other? It's definitely not a trivial problem. Actually, it took many, many years of algorithmic study. And finally, the first paper was published in 1995 by Pavel Pevsner and Sridhar Hanni-Halley. And they found a polynomial time algorithm that can actually tells you how to sort one into the other. And if you don't really care about the sorting sequence inversion, you just want to know the number of inversions, actually, there exists an algorithm that allows you to compute the distance in linear time. So it's complicated, but it's doable. It's very efficient. Whereas, here's another thing that's interesting. The inversion distance here defined is for gene orders that have the sign information. You know which of the 2 strands each is on. If you drop the sign information, the problem becomes NP-hard. For transposition distance, that's not a totally different matter. Actually, no one knows how to compute this exactly other than just brute force trying to do that. And we don't really know if it's an NP-hard problem or not. It's one of the open problems in gene arrangements. The third distance people are looking at is called the breakpoint distance. It's a very simple distance that actually is surprisingly informative. So look at these 2 gene orders, example gene orders. A breakpoint is a place where the gene order changes. So I highlighted them using the red vertical lines. So for example, gene 3 is followed by gene 4 in A, but gene 3 is followed by -8 in B. So the gene order breaks at that point. So that's a breakpoint distance. And the breakpoint distance is just the number of breakpoints in one gene order with respect to the other. And this is a symmetric measure. You can swap A and B and you get the same number of breakpoints. And all 3, of course, by their definition, they are all metrics. They satisfy triangular inequality. They're 0 if and only if the 2 gene orders are the same, and so on. And obviously, it's really easy to compute. So these are 3 kinds of distances that people study. Yes?
Transposition, again, if you take 2 contiguous pieces of arbitrary length.
Yes. Yes, you swap these 2. They have the same effects. If it's just unsigned transposition. If it's signed, then one of them has to be reversed. You need this to be able to generate all possible genomes.
Fix the length of the interval.
Yes. Yeah, I think if it's 1, then you can sort it, right? But for 2 or more, I don't know the answer. Yeah, but that's a very good point. There are approximation algorithms for transposition. You're absolutely right. But how to compute it exactly, we don't know. And actually, I'm going to show you that it's really absolutely important to compute these distances as accurate as possible to get the tree right. Okay, so we have the distances for genome rearrangement evolution. We also need to know how these events happen. What is the relative probability? Which kind of events happen more frequently than the others? The model we're going to focus is called the Nadeau-Taylor model. It's proposed by Joe Nadeau and Ben Taylor in a PNAS paper in the mid-'80s. So what they did is they look at the syntactic regions between human and mouse genomes and see, well, what is the distribution of these lengths? What they found is if they assume the breakpoints, the endpoints of these, the breakpoints are distributed uniformly across the chromosome, then they can explain the distribution of the syntactic region lengths really reasonably. So we call these Nadeau-Taylor models. They're uniform models if the endpoints happen uniformly random across the gene order. So what it means is whenever inversion happens, you choose two possible breakpoints between any two pair of genes randomly, and then you apply the inversion. And the same thing with transposition. Because it's the same as swapping two segments, that means you have to choose three endpoints. And that means all inversions are equally probable. And it turns out that it's not entirely true for human mouse nuclear genomes. There are recent studies, primarily by Pafner, that shows that if you have better resolution maps, actually there seems to be hotspots in breakpoints. And also David Sankoff shows that for bacterial rearrangements, really short inversions seem to be really probable. But for plant corpus, it seems to be working really well. So we're sticking with this model. But this model only assumes there's one type of event. So we want to look at inversion and transposition simultaneously. We want to have a model so that when you study any results of these models, they can be applied to both cases. So what we came up is, well, we can put them together as a mixture model. And within each of the three classes, they have the same probability occurring. They're still uniform, but there's a fixed ratio saying which class is more frequent than the others. So you can just adjust these parameters, say, then you have inversion-only model, you have transposition-only model, or you could have all of them. And any result that's based on this model can be applied to all these cases. So now, yes?
It seems that under these models, you could compute the distance just by looking at the breakpoint distance, because the chance that you'll have a collision where the same breakpoint occurs in two inversions of two operations, unless there's about square root of n of them,
Yes.
will be small.
Mm-hmm.
And so it seems,
Exactly, yes.
So it seems that you could just look at the breakpoint count and it would tell you how many operations.
Yes. That's a very important observation. Actually, some of my results are based on that observation that leads to approximations. And one thing that's interesting is breakpoint gives you these information, but not all the results. And actually, inversion tends to be more accurate than breakpoints because it keeps more information. I'll show you during the using a simulation. That's the case. Now we have the Additive Distance. We have the model for rearrangements. We want to look at what kind of methods are available for tree reconstruction. Here I roughly divide them into just two categories. It's a very rude classification, but it serves my purpose for the talk here. The first class consists of distance-based methods. Most of them are very, very efficient. It takes only seconds to run large data sets. What they take is the input is a distance matrix. You give it a pairwise dissimilarity, for example, inversion distance or the breakpoint distance, and then you try to build a tree that best matches this distance matrix. So that, of course, means you want to have a really good distance estimator that reflects the amount of evolution between any two gene orders. And there are many good methods that actually give really good performance. Neighbor joining, one of the most popular methods, and my impression is probably the second most cited bioinformatics methods paper, only after BLAST. That's my impression. And Weber is a variant to neighbor joining that uses not only the distance estimator, but the variance of the distance estimator. So it tries to minimize the overall variance when doing the algorithms and tries to improve the accuracy. And it seems in their paper, they actually have better performance when compared with neighbor joining for DNA sequences. And FastME is the latest method that I've been using. It's very fast tree search algorithm for distance-based methods. It actually has very good performance. But I'm going to focus on neighbor joining today. The second class are what I call, these are heuristics for NP-hard optimization problems. And I put maximum parsimony and maximum likelihood in this class and Bayesian methods. That's another approach that people are using for gene rearrangements. The leader for that approach is Brett Loggett at Wisconsin. But I'm not going to talk about that today. Today, instead, I'm going to focus on maximum parsimony. So the difference between this and this is this actually works on the sequence data for DNA, in this case, gene order data directly. It's not based on distance. And usually, generally speaking, it needs to search through the tree space, try to score as many trees as possible, and find which tree that gives you the best score, for example, lowest parsimony score. And that, of course, means the size of tree space cannot be too large if you want to really search for the tree space. And, of course, the size of tree space grows exponentially as the number of leaves increases. So even with 50 leaves, the number of trees is about 10 to the 74, which is pretty close to a number of atoms in the universe, 10 to the 80. So, but fortunately, there are a lot of efficient tree space search algorithms that can give you really good performance. So our first attempt is we're going to use distance-based tree reconstruction. The overall workflow is like this. You start with the input gene orders. You compute some kind of distance estimator that estimates the amount of evolution. And then you apply your favorite tree reconstruction method, and that gives you a tree. In the Phylogenetics community, usually the performance of these tree reconstruction approaches are studied by simulation. So what we do is we first generate a model tree, either using a biological tree, estimate of real data set from literature, or just generate some model trees based on some theoretical distribution. And then we simulate the evolution process along the tree to get a synthetic data, say, of course, the generalized Nadeau-Taylor model. And now, you have the tree, so you can compare these two trees and say, well, how similar the inferred tree is to the actual tree. And that tells you how good your method is. There are many different measures we can use. We're going to use the Robinson-Fultz distance, the false negatives, which is based on Robinson-Fultz distance that Luai was mentioning in his talk earlier. The first approach were both done by David Sankoff's group in the late 90s. They tried two approaches. One is the distance-based approach, which is just using neighbor joining together with breakpoint distance. And they studied the animal mitochondria data set for that. And also they studied a smaller data set using a maximum parsimony formulation called BP analysis, which I'll talk about later. The idea is to find a maximum parsimony solution using breakpoint distance. The BP analysis is really the name of their software, and it tries to search through all the tree space exhaustively. So in their paper, they only handle a 6-gene order data set. And later, the method was improved after many years of efforts by the team at University of New Mexico. The leader is Professor Bernard Murray, now at Switzerland. The software is called GRAPPA, and my impression is still the most accurate maximum parsimony method for gene rearrangements. This is a simulation study we did to see how accurate these methods are. In this case, we're looking at 40 gene orders, 120 genes, number of genes in chloroplast, and we assume inversions and transpositions all occur with equal probability. And we generate the model trees using the U-Harding process, and then we perturb the branch lengths a little bit, so it's not totally ultrametric. Here is the results. The x-axis is the diameter of the input data sets. That is the maximum pairwise inversion distance between all 40 gene orders. So it is an indicator for the amount of evolution in your data set. The higher it is, the more events there are, and usually the more difficult it is to get back the tree. The y-axis is the false negative rate. That is the fraction of edges in the inferred tree in the model tree that we failed to infer. So these are edges we lost. We didn't infer. So our goal is, well, we want to achieve a 10% error. We'll be happy with that. And this is the performance using neighbor joining using breakpoint distance. For BP analysis, it will not finish because 40 tags is really, and 120 genes is really too much for the program. So we could only test neighbor genes using breakpoints. You notice that for small data diameters, it works pretty well. It almost gets the whole tree back. But when the diameter goes up, and for the highest setting, the error can be as high as about 40%, which is clearly not acceptable. So let's step back a little bit and look at the big picture of this. So in this illustration, the x-axis is either the problem size or the diameter of the data set that indicates how difficult the data set is. And the y-axis is the error. The lower, the better. First, we have neighbor journey using breakpoint distance. You can apply it to any data set we have. It's very efficient, but the accuracy leaves something to be desired. Breakpoint, BP analysis or GRAPPA, these are exhaustive methods for maximum parsimony. They can only handle really small data sets, but they are very accurate. So what we want is, of course, something that's in this region. We want a method that can handle large data sets, but gives you good performance. So we're going to take several approaches to try to solve large data sets. The first approach is, of course, going back to a distance-based method because they can handle large data sets. There are many good phylogenetics reconstruction methods for that, so we're going to focus on distance estimation. What is the best distance estimator that can give you more accurate trees instead of just breakpoint distance? The whole concept of distance-based phylogeny can be traced back to the concept of true evolutionary distances, or we call we can also call them additive distance. The idea is this. If somehow here we have this tree, and the branch length is the actual number of rearrangement events, the actual number of events that happen between them. So this records the whole history of the evolutionary history of the different species. And if you look at this red line, you can compute the distance between them by adding up the branch lengths. That gives you a total actual number of events that happen between these two. So there's a theorem that's proven by Mike Waterman in the 70s that shows that if you actually have this distance matrix in this form, the additive distance matrix, you can actually get back to the tree with 100% accuracy if the distance is exactly corresponds to the past distance on the tree. Well, in reality, that's not going to happen. We don't have the tree topology, and evolution is random, so you could have all kinds of problems like homoplasy, randomness, and so on. So, and also, we don't have tree topology, so what you're estimating is some minimum edit distance between the two gene orders. So the question is, well, that's the only information we have. How can we get back to this, how long this red line is, given the minimum edit distance? So that's the concept of distance correction. For example, juke scanner in DNA sequences. So that's what I'm going to do. If you look at some simulation data to illustrate the point, here what we do is we take a gene order, we apply random inversions to it, we get another gene order, then we compute distance between them. And that is the estimated minimum edit distance, that's the x-axis, and the y-axis is the actual distance, number of events. These are the straight lines that corresponds to the actual number of events is the same as what you estimate. The breakpoint here is because the way we compute breakpoint distance is actually every inversion induces 2 breakpoints. So the slope should be divided by 2. So what you can notice is the linearity saying that the edit distance is the same as the actual number of events only works when you have a very small number of events. When it increases, it gradually deviates from the 45-degree diagonal. The range of linearity, of course, it seems that inversion is higher than the breakpoint distance. So what that implies is if you use inversion distance, probably gives you a more accurate tree than breakpoint distance. That's what it implies. But eventually, they all max out to the maximum allowed edit distance, which is number of genes in both cases. So what we can do is, well, let's figure out what these curves look like, and then we do some adjustment by a lookup on the curve, which is the same as computing the inverse of this curve. So what is this curve? That is no different from the expected breakpoint or inversion distance as a function of the actual number of inversions. So that's what we're going to find out. And you can use breakpoint or inversion distance and do this distance correction idea And we have 3 different distances. They have slightly different derivations, but they all have the same goal, how to convert the minimum distance to the actual number of events. So the first we're going to look at is the breakpoint distance because it's easier to handle. So if you think of it more closely, the whole problem is no different from we're going to study a Markov model where the state space is the state of all possible genome arrangements. And of course, that's too big for anyone to do the actual analysis. It's very unwieldy. It's hard to manipulate. But since we're finding the expected breakpoint distance, by linearity, we can look at individual breakpoints. So that makes the problem a lot simpler. So by the uniform model, all we need to look at is, well, whether gene 1 and gene 2 are adjacent at any point. And then we just multiply by n because it's symmetric. So that reduces the whole problem into a Markov chain with 2 times n minus 1 different states, which is, of course, now you can actually compute that. So what you do is, of course, you find out the transitional probability matrix. And there are simple closed form formulas for you to do that for inversions, transposition, inverted transpositions. By the mixture model, all you need to do is to just do the weighted addition of the 3 matrices. That gives you the overall transition probability matrix. What is even better is actually people have figured out how to find out the eigenvalues for inversion-only matrix, but not for transposition matrices. So you can actually compute this, the expected breakpoint distance really quickly using these eigenvalues. It's done by Nicholas Eriksson, now at Chalmers, Sweden. So that is the first attempt. We look at the Markov process of breakpoints. Well, there's something else you can do, which is, well, why don't we just lump all these breakpoint states, 1 and 2 are not adjacent together, into a single state. There's a breakpoint. And look at the probability of between breakpoint and no breakpoint. It is not a Markov chain, however, because the probability from jumping from 1 to adjacent to a breakpoint is the same of all these. But jumping back actually is different. It depends on where gene 2 is with respect to gene 1 because the number of inversions is slightly different. So, but what we're able to do is actually, well, these are constant, these are variable, not constants, but we can find lower and upper bounds to this parameter u, meaning canceling a breakpoint. So that gives us 2 Markov chains with 2 states each. And you can compute these using a closed form formula really easily, and that gives you lower and upper bounds for the breakpoints. And the good news is this lower and upper bound is very tight. It's inversely proportional to the number of genes. So for 120 genes, it's actually not bad. It's very accurate. The accuracy to the actual expected breakpoint distance is within 1 breakpoint. There's another thing you can do, which is you can actually look at all these breakpoints simultaneously. The breakpoints themselves are not independent at all. So the idea is, if they're independent, you're going to have a non-zero probability of having just 1 breakpoint. But it never happens. So it's not dependent. So although for expectation, it's okay to look at 1 breakpoint. If you want to look at higher order statistics like variance, you need to look at all the breakpoints simultaneously. So we came up with this idea of, well, why don't we take the advantage of that when a breakpoint happens, it's really hard to get back, like what Dick was saying. So that is the key observation here. So we came up with this box model for approximation. Initially, we have n empty boxes, assuming there are no breakpoints. And when you have an inversion, you choose the 2 endpoints and you put a ball into that if there is one. If there is, you don't do anything because it's a breakpoint. It's really hard to cancel that. And what you can do is you can actually compute all the moments for this model, for the number of non-empty boxes. And that particularly gives you the variance of the breakpoint. And you can plug it in to the Weber methods. And actually, that improves the accuracy than just using neighbor joining. The third distance is inversion-based distance. The problem is inversion distance is a lot more complicated than breakpoint distance, although it seems that inversion distance is more informative based on the simulation we just looked at. So what we did is we just do numerical simulations. Gives us scatter points between inversion and the actual inversions, and we use a numerical method to find out what is the best fit to that. And then we're going to use that to do our distance correction. So if you do these distance corrections, that's what you get. The actual number of inversions is going to be almost the same as the corrected distance, even up to many inversions. Although, of course, at that point, the variance is higher. And if you you use this distance for tree construction, it improves the actually a lot. This is the red line is the neighbor joint using breakpoint, and the blue line is the one that's using the new corrected distance. And you have now the method returns the error is 10% or less, only except for the highest settings. Well, that's the first attempt, distance-based methods. The second attempt is, well, we want to solve it using maximum parsimony. So like DNA sequence, we can define the maximum parsimony problem for gene arrangements basically like this. If you look at any particular tree topology, the internal nodes are open. We don't know what the gene orders they are. But what you want to do is to find the two optimal gene orders such that if you put them here, the total edge length, the length of the tree, is minimized. So that is going to be the score of the length of this tree. And the overall problem of gene arrangement parsimony is to find a tree topology or tree topologies that have the shortest length. This is actually a very challenging problem compared with DNA sequence parsimony, where if you have the tree, you can compute the length in linear time. This problem is NP hard, even for the simplest problem with only 3 gene orders and just one node that's at middle. You want to find the optimal median. It's an NP hard problem, although you can reduce the breakpoint median to a TSB problem, and that's what Sankoff and Blanchett did in their BP analysis. And you can also solve the inversion median in the that's implemented in the Grappa software. And then with the median, what you can do is this. You have a tree, you have the gene orders at the leaves. You initially assign some gene orders as initialization, and then you run the median of 3 problems repetitively. Every time you solve the median of 3 and you update this gene order. You keep doing that until you can no longer do refinements. And at that moment, that is the best score you can find for this tree. So there's no guarantee this is the optimal tree length. But hopefully that's going to be a good estimate. Again, that is very time consuming. You're running tons of NP-hard problems just to score one tree. So we came up with this idea that transform the gene order like this into an encoding that's just like DNA sequence matrix. So what you can do is you look at every gene and look at what gene follows that. So in this case, 1 is followed by 2 in A, followed by -4 in B, and you need both directions to have complete information to reconstruct the gene order. And then you end up with a matrix that's just like DNA sequence with numbers. The good thing about that is now you can feed this as input to your DNA sequence software and solve the parsimony using DNA sequence algorithm that is linear. So it's very fast, and you can prove it's optimal under this setting. One thing to think about this, it's really a relaxation of the original gene order because the internal nodes don't have to be gene orders, but they're these kind of encodings that do not necessarily correspond to gene orders. And also, you can prove that it's a lower bound to the breakpoint distance, the Hamming distance in this setting. So if you actually do that, again, it's better than distance-based methods. And another thing good about it is you can actually handle larger data sets, whereas Grappa and BP analysis cannot because they are larger. And again, this problem takes a lot more time, like 10 minutes. It's as opposed to distance-based methods usually take seconds. But and there's another limitation. It's more of an implementation issue, which is we're using Pulp, very popular software for phylogeny to do this. And Pulp only allows 32 different states for every site. So if you have 100 taxa, everyone gives you a different gene order, then Pulp cannot handle that. So that's an implementation issue, but in practice it's very important. So there's a limit to the size of data set you can handle. So if you look at, here's the real data sets that I'm going to talk about. This is the Bellflower family that we studied. The data set consists of 12 Campanulaceae species with tobacco as outgroup. And each one has 105 markers. Some of them correspond to 2 genes, for example. And one particular characteristic of this data is actually has a higher than usual rearrangement rates than the rest of the plant chloroplasts. So that's why it got our collaborators interested in the first place. We don't know the actual trees because we don't have gene sequences. So all we can do is we compute the inversion length for the trees you get using different methods. The best one is grappa using inversion distance. That's the exhaustive method using inversion. If you use breakpoints, it's only one extra inversion. But the running time is actually a lot because grappa has some bounding methods to eliminate tree search, the number of trees to score in the tree search. And inversion seems to have a better, tighter lower bound that allows you to do more efficient tree pruning. If you use NPME, actually it only takes 10 minutes as opposed to hours, hundreds of hours. But NPME actually has the same performance as grappa BP. Here's another thing that's interesting. So initially when they were looking at our biologist collaborator, Bob Jansen, we're looking at this data set. They're trying to do the scenario by hand, and they found that they had to use 2 transpositions, which they're not very happy because almost all the other events are inversion. And our software can actually solve the scenario using just inversions, actually with fewer rearrangement events. So that shows that for chloroplasts, it seems that inversion is enough to explain the data set. So how much more time do we have? 12 minutes, okay. So at this moment, we have this. We have a more accurate distance estimator, gives you more accurate trees. Grappa is the most accurate method, but only for small data sets. NPME can handle larger data sets. So we're still not there. So what we're going to do is we're going to use a divide and conquer approach to combine these 2 methods, NPME and neighbor joining. The idea is this. If somehow we can figure out a way to divide the original data sets into overlapping smaller data sets and then solve each one, now these are smaller, so it's more accurate and you can actually handle them. It's not too large, so NPME fails. And then you put them together using supertree methods. So what it means is, well, first we have to have a good guide tree, which is the initial starting tree to tell you how to partition the data set. Well, for that, we can use neighbor joining using ED distance because that's most accurate. And we have to have small subproblems, not only because the diameter is smaller means it's more accurate, but we cannot let it to be too large, so NPME cannot solve that. So we have an algorithm that does that. Actually, it gives, you're allowed to specify an upper bound to the size of the subproblems so that it's never too large. If it's less than 32, then every gene is only followed by 32 different genes at most, so NPME can always handle these data sets. So if you do that, simulation shows that this is a larger simulation setting than the previous one. The red rectangle one corresponds to the diameter settings for the previous. And in this case, we're looking at 200 taxa, where for almost all the other methods, they cannot handle these data sets, only the distance-based methods. And if we combine the 2 methods using this divide and conquer methods, it's always about 10% lower in terms of the error. So this method can handle larger data sets. It has better performance than distance-based methods. So at the end, I'd like to talk about this another approach that we tried, which is really interesting. It reflects the complexity of genome rearrangement phylogeny. It's what I call global versus local search. So if you let me remind you that what grappa does is it's a series of local iterative refinement that try to find optimal medians for internal nodes. So at any time, you only look at the 3 neighboring gene orders. It's a very local information. Whereas NPME is a relaxed version that uses breakpoint distance. And the good thing about this is actually it gives you a global optimization for the internal nodes. It uses all the information simultaneously, although it's a relaxed version. So it got us thinking, well, can we combine these 2 methods and see if it gives us more accurate trees? So that's what we try to do. So the idea is this. For every tree, when we want to compute the score, first we compute the optimal NPME, which is like DNA sequence, so we can figure out the optimal sequences for the internal nodes, which do not necessarily correspond to gene orders. And then we're going to translate each of the internal node sequences to the closest gene order. And then we can continue with the grappa iterative refinement procedure. Then we end up with the length of tree. So we hope that by using this kind of global information, it can give us better tree length estimation. It turns out that it's not, at least we haven't figured out what is the best way to do that. If you have an NPME sequence that does not necessarily correspond to a gene order, the closest gene order, which is the gene order that its NPME sequence is the closest to the input in terms of breakpoint distances. If it's unsigned, you can show it's NP-hard, but if it's signed version, we don't really know if it's NP-hard or not. And so we came up with a greedy algorithm heuristic to do that. So that allows us to implement the whole algorithm. So we have two questions. Is it a good median solver? And does that give you more accurate trees? So this is the simulation study. We try to generate a lot of synthetic median of 3 data sets. And we look at the median solvers, the new median solver using NPME, the old breakpoint inversion median solvers, how different they are from the actual median. So the larger the distance, the worse the median solver. So what you can see here is, no, it's not a good median solver. After all, it's a relaxed version using breakpoint. Inversion is probably the better distance. And in some scenarios, it's actually pretty bad. It's never as good as the more sophisticated breakpoint inversion medians. But somehow, if we put it into the tree reconstruction, it gives improvement. This is the length of the best tree normalized by the new NPME grappa algorithm. So 100% means it's the same. And so that's this. We use NPME, which is breakpoint based. Then we use inversion median solver to find the tree length. If you don't use NPME, the best one is, well, inversion median. And the difference is really small, less than 1% in terms of tree length. But if you look at the accuracy of trees, it actually gives you a very big difference. Whereas the new method is about 2% error, 3% error in 2 settings. If you don't use the NPME, global optimization method, the inversion median actually is about 3 times higher in terms of error. So what it says to us is, well, first, the new method is not a median, but that's not what we want. But even a naive optimization that uses global information actually does better than really sophisticated optimizations that's iterative but local in nature. So, and also, even a small tree length improvement leads to a big difference in the tree topology, in particular in the gene rearrangement phylogeny. So what this means is you need to get the tree length and distance estimation as accurate as possible. And of course, that's not a trivial problem. So that's the conclusion, the summary of the talk. I've talked about different kinds of approaches to scale the gene rearrangement phylogeny so you can handle larger data sets. And one thing I didn't mention is all these methods are robust to model misspecifications. So what it means is some methods require to specify the mixture probability. And if you get it wrong, it still gives you really good performance. And of course, the next step is really to generalize all these results to more complicated situations where you have non-uniform models, like you can have hotspots, you can have short inversions that happen more frequently, and you need to be able to deal with insertions, deletions, and fusions, translocations that gives you different gene contents and multiple chromosomes, so you can actually work with nuclear genomes. So acknowledgements, and I have a lot of collaborators. Tenny Warno's my advisor, and Bob Jensen's the biologist who gave us the data sets. Bernard Murray is the PI for the grappa software, and now he's at Switzerland, and with Ji Jun, the principal programmer for grappa. Now he's at University of South Carolina. So thank you.
Questions?
Yes. Like bootstraps, for example? That's a good question. We never tried to do this, bootstrap, but that's, well, bootstraps, for DNA sequence, you need the assumption is the sites are evolving independently. But that is not the case for genome arrangement, where the breakpoints are independent. So I guess one thing you can do is say a Bremer support. That's something that you don't need this kind of independence, but you can still, yes? That's true. That's true. Mm-hmm. Yeah, parametric bootstrap. Yes?
So if we're adding gene duplications and hotspots, how difficult would it be to apply this method?
It's a lot more difficult, especially with duplications. For example, the breakpoints, when you have, say, two genes, two copies of the same gene, the breakpoint distance itself is not well defined. You can define some kind of minimal matching, but it's not a metric anymore. And it's not trivial to compute. So with inversion, well, there's no algorithm for computing inversion between genomes with duplicate genes. So it's a lot more complicated.
Any other question? Let's thank the speaker.
Thank you.
So the final speaker of the morning is John Holsenbeck from UC Berkeley, and he will be speaking on Bayesian inference phylogeny, of phylogeny.
Right, or I changed the title at the last moment. So first of all, can everybody hear me properly? Is this microphone working? It is not on?
I hear you, but I don't think it's on.
Well,
The question is, is it getting recorded?
okay, good.
It's more for the recording than for us.
Yeah, you guys don't matter, just as long as the people out there. So I did change the title a little bit. It's now Phylogenetics in Chinese Restaurants. You'll see eventually, I hope, if I have time. And I've always taken it as granted that everybody knew who that guy was on the slide, but I recently gave a guest lecture in a graduate course in bioengineering at UC Berkeley, and I asked the graduate students who this guy was, and I got Boltzmann, Maxwell. But nobody could actually guess who it was. It was some 19th century scientist. Yeah, you're right. That's Darwin, of course, Charles Darwin. And so, of course, he had a couple big ideas. One, actually, this idea predates him by a bit, but that all species on Earth are related to each other by common ancestry. It's the tree of life that we've heard so much about in the symposium. And then he also was able to explain why there's a match between organisms and the environment. He explained that with natural selection. I'll be touching on both of those things briefly in this. And we've heard a lot about the phylogeny problem. Simply put, it's just how can we estimate the genealogy of a group of species. And Joe yesterday gave a lot of good reasons why you might be interested. Here's my little list of reasons you should care. And I just told you it's a central idea in biology. There's a lot of interest in the evolutionary history of life. But we haven't really heard much about host-parasite co-speciation, but phylogenies are used as evidence for that. There's a lot of practical importance of phylogenies in the field of epidemiology. And of course, there's a lot of interest now in trying to decipher how the genome evolved, and phylogenies are playing a great role there, and I guess molecular evolution's related. And I also just think there's some kind of interesting results. We now know if you're interested in genealogy, my great-grandmother liked to sort of trace her genealogy back, and if she traced it far enough, she would have found that the chimp is our closest relative, and I suspect she would not have liked that conclusion, but it's true. But there's other things you may not be aware of, like birds are dinosaurs. They descended from dinosaurs, so dinosaurs in one sense aren't extinct. The birds are the living relatives or descendants, and that if you ever eat a king crab, it's actually nested within hermit crabs, which I think is kind of cool. So anyway, so phylogenies have been very useful, and here's an example I've given many times, but this goes back now well over 15 years, and perhaps I had to come up with a better example. But about 15 years ago, there was this question about a dentist in Florida who had a lot of patients that mysteriously became infected with HIV, and there was some concern that perhaps somehow he was intentionally infecting his patients. And so what they did is they did have viral sequences sampled from the patients, and you can see the patient sequences here. And they also had some of the viral samples from the dentist, and they sequenced a particular gene from the virus from the dentist. They also had sequenced a bunch of people from Florida that never visited this dentist that had HIV, so there's the so-called local controls. And the point is that many of these patient sequences clustered with the dentist, and that was taken as evidence that perhaps the guy was dastardly and somehow infected his patients with the virus. And there were a couple patients you see that don't cluster, but later these patients were found to have had other risk factors. So anyway, so phylogenies are important. We're all in agreement, I hope. So I want to talk a little bit about the modeling assumptions we make and some of the interesting questions I think we can address in phylogeny. And basically, there's two parts to a phylogenetic model. You're probably aware of that by now. But we have a tree. That's the tree that, yes?
Sorry, before we...
Oh.
So what was the conclusion of
Oh, so the dentist was a bad guy. That's the conclusion. I would not want him as my dentist. That was another conclusion I had. What's that?
He wants
No,
to know if
so
it was
that's actually, so it's a, just remember this if I go over time. But no, the dentist actually died before they could do anything about it. But it turns out that there was another dentist. It's something about dentists, I'm worried. In Louisiana, I believe, who infected his girlfriend with HIV and, just for reassurance, I guess, hepatitis. And they used the same sort of techniques, actually, phylogenetic techniques, and it was admissible in a court of law. And so this is a PNAS paper. I remember David Hillis was one of the authors on that paper. But basically now phylogenies are there's precedent, right? I guess that's important to lawyers for some reason. And this kind of makes it, just as an aside, it's imagine yourself sitting in jail based on the UPGMA or something like that. That sort of scares me. At least I want to be convicted on a Bayesian analysis or maximum likelihood or something like that. Okay, so anyways, that's no more dentist stuff, and just be careful of your dentist. Don't brush your teeth. That's my main message here. Anyway, so we know that the parts of a phylogenetic, the things you put into the model. One is a tree with branch lengths, usually measured in terms of expected number of substitutions per site, and we know how interesting that is. But there's also this sort of neglected bit down here, which is we have to have some sort of mechanism describing how the characters, whatever they might be, DNA sequences, amino acids, whatever, evolve along the tree, right? We need to specify both. And it's usually the case, and I think most of the talks we've seen here, most people seem to be interested in the phylogeny, and in a sense it's like, well, if we could just do away with this, that would be great. So it's sort of considered a nuisance parameter in the analysis. And there's lots of ways statisticians deal with nuisance parameters in analysis. Well, all the analyses you'll see here, the tree is the nuisance parameter. I have to have a tree because the sequences are related to one another. But I'm going to be concentrating on the mechanism of character change and asking us what that tells us about the evolutionary process. Okay? So it's a phylogenetic talk, but you won't see many trees because they're nuisance parameters. I'm integrating them out of the problem as much as I can. So the first thing I want to talk about is just how biologists go about choosing models of character change. And there's actually quite a bit of sophistication, I think, in the field. They basically will do a likelihood ratio test for nested models. You heard a little bit about likelihood ratio tests. They can use the Cocky information criterion. It's usually the case that there's a small set of candidate models that they consider, Juice Canner, Kimura, Tamurine, GTR. That's usually about it, right? For instance, for DNA sequence models. And there's even fewer if you go out to amino acids and so forth. And there's even been some programs that sort of automate the selection procedure, like the program model test is almost seems to be a prerequisite now for getting a paper published in a lot of journals. So I'm going to describe a different approach. It's basically the first thing I'm going to do is I'm going to be expanding the pool of candidate models. So I'm going to be considering all models of a certain class. And I'll be interested in calculating the joint posterior probability of the models and the other tree parameters. So I'm going to be throwing everything into a Bayesian analysis in this case. And I'm going to be using Bayes factors to evaluate the support for various hypotheses. So there's a couple of things you may not be aware of, but the first thing I'll do is just quickly describe Bayes factors. Bayes factors are very simple. They're the ratio of the marginal probabilities of the data. I always use data for X for some reason, so an alignment in our case. Conditional on model 1 divided by the marginal probability of model 2. Marginal likelihood of model 2. Okay? That's what a Bayes factor is. And basically, if the ratio is greater than 1, that means model 1 makes the data more probable. If the ratio is less than 1, it makes model 2 more probable. Very simple, right? So it's a way of evaluating the support for models. And they're related to likelihood ratio tests that you saw earlier, except you're marginalizing over the parameters instead of maximizing the likelihood with respect to them. And you can also calculate the Bayes factor as the posterior odds to the prior odds, and I gave an example here of how you could actually do that if you had a posterior probability of one hypothesis of 0.83 and the prior probability is 0.03. Well, then the posterior odds would be 0.83 divided by 0.17. The prior odds would be 0.03 divided by 0.97. There's ways of interpreting, like I'd already told you about the ratio of the marginal likelihoods, but you can also think about this as the degree by which you change your mind and moving from your prior beliefs to your updated beliefs, right? And there's even tables. This is something that takes a lot of people some getting used to if you've been grew up on p-values and confidence intervals. Well, you won't see any p-values or in Bayes factors. So basically, you have to sort of evaluate those numbers on their own terms. All right, so that's first. Just a little bit about Bayes factors. Now, the first thing I want to talk about is how I've been interested in expanding the candidates.
There's no sort of objective number you can assign to it. It has 90% probability.
No. So we saw a little bit about predictive p-values yesterday in Vladimir's talk, and I've always wondered about those because even though I've used them, it seems like they're trying to get their p-values as well as doing something Bayesian. It seemed like cheating to me to some extent, but I'm perfectly willing to use it if people like their p-values. You can get things that are sort of analogous, but they're not really p-values. So the first observation I'm going to make is that in terms of a lot of the that's not a universal statement, but for many phylogenetic models, I think you can actually describe the different possible models as partitions. And I mean partitions in the mathematical sense, where a partition is a set of distinct objects. A partition of a set of distinct objects, S, is a set of disjoint subsets of S whose union is S. So just to give you some examples, here's the most general time reversible model that most people are interested in. There's two sets of parameters here. One is these pi parameters. These are the stationary frequencies of the continuous time Markov model. And most phylogenetic models have this unusual property that the stationary frequency parameters are built right into the rate matrix, but that's fine. And then they have these rate parameters, which have R with some subscript. And these are often sometimes called exchangeability parameters. And so I'm going to be concentrating on the exchangeability parameters here, these R parameters. Just to remind you, this rate matrix specifies a rate of substitution from one nucleotide to another. So it's an instantaneous rate matrix. So here's one possible partition of those rate parameters I talked about. I just laid them out here, RAC, RAG, and so forth. And this is a description of a partition, which many people use. It's called the restricted growth function notation for a partition. But basically, the first element always gets the index 1. The second element gets the index 1 if it's in the same subset as the first, and so forth. So here's a case where all the rates are in the same subset. They're all the same. And that would correspond to either the Jukes-Kantor model or the Joe Felsenstein 1981 model if you have potentially different stationary frequencies. So there's one model. So here's another model you can have. This is one where you have the AG rate and the CT rate. These AG is called transitions, confusingly. And then the other possible rates, these are the four rates, the ones that are indexed 1 here are called transversions in the evolutionary biology literature. But you're basically pulling out two of the rates. And that gives you either the Kimura model or the so-called HKY 85 model, the Hasegawa-Kishino-Yano model. Here's another model where you basically partition this third, the other, you have two independently estimated transitions. That's the Tamura model. Here's an unusual model. This is one where the AG rate and the AT rate are both constrained to be the same. Anybody know which model that is? Well, I decided to name that one because it's unnamed. That's my daughter, Beth. So this is Beth's model. So there's enough models. There's not enough for everybody, but there's a lot of them. And so I figured she's a very nice girl. She gets her own model. And then, of course, you could have the GTR models would be indexed 1, 2, 3, 4, 5, 6, okay, under this scheme. So you can ask, what's the combinatorics? How many models now are there? Well, there's 203 of them. One of them, of course, has one substitution type. There's the index. One of them has 6 substitution types. That's the GTR. But you have 31 with 2 substitution rates. There's our Kimura model. 90 with 3, 65 with 4, and 15 with 5 substitution classes. And if you wanted to know just the total number of models, what's the Bell number? So the number of models with K subsets is given by the Stirling numbers of the second kind, and the Bell numbers gives you the total number of models. And like many of these things we see in phylogenetics, the Bell numbers grows very quickly. So for 6 subsets or 6 elements like we have here, we have 203 possible models. And actually, I've been interested more recently on amino acid models. And so you can have an amino acid model that's isomorphic to the GTR model, where you have 190 possible rates, right? In which case you have like 10 to the 258 possible models, 6.59 times 10 to the 258 possible models.
Really have a lot of kids in that case.
And then everybody gets the model named after them, right? And so if you're disappointed about not having a DNA sequence model named after you, there's hope. So that's anyway. So I'm just telling you we could describe the models in a pretty general way. So what I'm going to be doing is, so the next question we have is just how I'm going to be interested in calculating the joint posterior probability of the substitution models, but as well as all the other model parameters, trees, branch lengths, base frequencies, rate variation across site type parameters, and so forth. And I'm going to be using Bayes factors again to evaluate the support for alternative models. So if we're going to be doing things in the Bayesian framework, then what we're going to be doing is calculating something called the posterior probability distribution of the parameters. So here is what we want to calculate. We're going to condition on the data, right? There's the data, my alignment right here. But we want to calculate the joint posterior probability distribution of the trees, the set of branch lengths on the trees. I'll skip that one for the time being. The base frequency parameters, the substitution rate parameters, and an index for the model, right? And then this alpha parameter, I'm going to be assuming that the rates, substitution rate at a given site's a gamma distributed random variable. So this is the shape parameter of this mean 1 gamma that I'll be integrating over for rates. And to get that, I need to, you have a likelihood. That's the probability of the data condition on all those parameters, what's called the likelihood. We have a prior, we're going to be Bayesians here, so we have to have a prior distribution on all these parameters. And then we have a normalizing constant, which is the marginal likelihood, and that's the thing that's going to involve a summation over all possible models and all possible trees and multidimensional integral over all possible combinations of parameter values. So it's hard to calculate.
But that drops out in your ratios.
Sorry?
That drops out in your base ratio.
It's going to drop out when we do the Markov chain Monte Carlo. We're going to be approximating. We're not going to do anything analytical here, but we're going to approximate this with a numerical method called Markov chain Monte Carlo. And the magic of MCMC is that it's called cancellation, it turns out. And this thing will cancel out, right? So we'll never actually be forced to calculate this marginal likelihood, which we can't calculate.
No, but in the end will calculate base ratio.
So if you know the prior ratio, if you know the prior probability of something and the posterior probability of something, and you're only interested in the Bayes factors, then yeah, you can actually calculate the Bayes factor. But remember, to get the posterior probability of something, we need to calculate that. So at some point, we're going to have to calculate it, and we're going to be numerically approximating it with MCMC. So MCMC, I'm glad we had the next slide up. So just in case you don't know, I mean, a lot of algorithmists think a lot in terms of optimization, and here I'm going to be using a method that's not optimizing, it's sampling. And it has a deep literature. It goes back to the 1950s. This is Edward Teller of the hydrogen bomb Teller guy. So it has roots in statistical physics. There were some modifications made to the method in the 1970s, and it's still being modified even more recently to sort of generalize it. But the basic idea is you have some parameter space. Maybe you have x and y, your parameters, or theta 1 and theta 2 perhaps. And the height of the surface here is the posterior probability for that combination of parameters, right? So I'm imagining right now that you actually knew what that surface looks like. Of course, we don't know what that surface looks like. We're trying to approximate it. So how does this MCMC work? Well, the idea is you're going to plop yourself somewhere down in this surface to start the chain. So here, we'll plop ourselves down randomly somewhere on that surface, and that's indicated by the red dot. You have some sort of proposal mechanism that's largely up to the programmer. Well, you have to adhere to some rules. That's going to stochastically change the current value and propose a new state. So here I've just came up with a proposal mechanism where you sort of drop a hula hoop over and center it on the current value of the parameter, right? And then we're going to randomly pick a new value for the parameter, propose a new value from within that circle, okay? So here, for instance, one possible thing we could have done is propose that blue dot. And generally speaking, a little bit of hand waving here, but generally speaking, if you propose a dot that's uphill, that is, it has a higher likelihood, then you automatically move there, right? So here we move from the red dot to the blue dot. Now the Markov chain would be centered on the blue dot, so the hula hoop would be moved and centered on the blue dot. And we're going to propose a new value from within that circle again. And here's an example where the proposed state is this green value, which is slightly downhill, at least by eye to me, it looks slightly downhill. And the probability of accepting the green dot as the next state of the Markov chain is the new height divided by the old height. And you might recognize the new height will be the posterior probability of the new value. The old height would be the posterior probability of the old value. And the important point is that the marginal likelihood that we couldn't calculate cancels out of here. You're going to have one value up here, f of x up here, and f of x down here. It's going to cancel out. And I should mention that the general strategy for MCMC is we're going to construct a Markov chain that has as a state space the parameters of the model that we're interested in, and a stationary distribution, which is the joint probability distribution of the parameters. That's the secret. And there's rules you can use that will make certain that if you don't screw up on your programming and whatnot, that you'll actually sample properly. So here's what you would imagine if you could iterate that process over and over again, proposing new values and accepting or rejecting the proposed states. And because you're always accepting values that are uphill and only sometimes accepting values of the proposed values that are downhill, generally speaking, you'll sort of explore areas of the parameter space that have high posterior probability. You won't be driven to the top and stop, but you'll sort of hang out in areas of high posterior probability. And what I've shown you here is there's lots of literature is very deep, but there's things such as you often will discard some fraction of the early samples taken as what's called the burn-in. Often the chain is thinned, so you only take samples you only record samples every so often and so forth. But the secret is this, is that if you can imagine, you see sort of I divided this parameter space up with grids, but imagine you just counted the fraction of the time the Markov chain stayed or dwelled in any one of these little grids. Well, a histogram of that would be a valid approximation of the posterior probability that we're trying to approximate. All right? So for trees, I won't. So for trees, for instance, we don't have hula hoops to center on trees. What you do is you want to perturb the tree in some small stochastic way. And so here's a proposal mechanism just to get your head around this that was proposed by Brett Largent and Don Simon. It basically is pick at random 3 contiguous branches, which I've highlighted in red, and their path length, the sum of the branch lengths for those 3 is m. You randomly contract or expand that path length proportionally, and then you snip off one of the 2 incident branches that's not on the path and randomly and uniformly reattach it along the path. So here I've snipped off the tree right here and reattached it down there, right? And that's just the proposal mechanism. It has so what you would do is you'd presumably know the likelihood or store the likelihood for this tree, calculate the likelihood for this tree. If this tree has a higher likelihood, in general, you'd accept it. I mean, there's some other factors in there that you have to account for, but that's the general idea. All right, yes? You do, just like with other problems, you could have multiple peaks in the surface, and it's a potential problem for MCMC just as it is for hill climbing methods. There's some tricks that you can use. For instance, there's this idea of heating chains. So you have multiple chains, one of which is the so-called cold chain, the one that's presumably exploring the space you're interested in, and then heated ones where they're exploring a flattened landscape of the parameters. And those are the ones that can more easily flip between peaks. But yeah, you can definitely have a problem. So I need to speed things up here. I've got a lot to go through. So just a little bit about some of the proposal mechanisms we use. We basically use split and merge moves to propose new models. So just to give you an example, a split move, if this is the current model with a partition described like this, you could actually split the second subset up and make a new group. And a merge move would be just the opposite. You might pick this subset, the subset index 2 and the subset index 3, and merge those together into one subset. Split and merge, if you just split and merge, is enough to get from one model to any other model in some number of steps. And you have to also pick rates, the rate parameters for these. So basically the rate for a split move, or a merge move rather, is just the average of the 2 rates that we had. And a split move takes one rate, R, U is a random number, uniform random number, and one of the splits is increased by that random number, the other subset is decreased by that number. But you notice that the average is still the old value. So there's some technicalities there which I won't have too much time to go through. But basically we constructed a Markov chain. We have different proposal mechanisms, some of which change the tree, some of which change base frequencies, some of which change the substitution model. And altogether all these proposal mechanisms allow us to explore the joint posterior probability distribution of all the parameters. So we looked at a number of data sets. This is just giving you an idea of the size of the alignments. We're conditioning on the alignments. We're assuming that they're fixed. So we had some that had 64 taxa and 1,620 sites, and some that were smaller. And we ran the chains for a long time, 10 million cycles of proposing and accepting or rejecting states. We discarded samples taken versus the first million cycles, ran multiple chains, and so forth. And we used a uniform prior on this 203 model. So each model had a uniform 1 over 203 prior probability of in this analysis. So here's the first set of analysis, just one way of summarizing the results. So just for kicks, we looked at which model would be selected with the Akaki information criterion. I have an index on the model. So the simplest model, the Jukes candidate, is indexed 1. And the most complicated, the GTR model, is labeled 203. And I don't know, I forget, the 15, I know that one is the Kimura model. That's the only one I remember. But there's a bunch of unnamed models here, but they're all numbered. And then here I've got the model that actually had the highest posterior probability. So model 168 for for this particular alignment had the highest posterior probability, and the posterior probability of that model was 0.74. Okay, and then here's the Bayes factor. I know the posterior probability, I know the prior probability. I can use the posterior odds divided by the prior odds to approximate the Bayes factor. So you get an idea of how much our mind changes when we observe the new data about that particular model. There's a number of interesting things you can do. Now remember, this is just looking at the model, so everything else we've integrated over. We're not conditioning on any particular tree or set of branch lengths or anything, right? They're being integrated out of the model. We're only looking at the marginal posterior probability distribution of the substitution models. One thing that's interesting you can do is you can order all the models from the model with the highest posterior probability to the lowest and start including them in a set starting with the model with the highest posterior probability until you have a cumulative probability of 0.95. That's not called a confidence interval because Bayesians don't call anything the same as frequentists, but it's called a credible set. And actually, Joe Felsenstein is the first one in his dissertation in 1968 to propose credible sets of trees. And he now completely disavows it all, but he was the first to do suggest Bayesian inference in evolutionary biology. But anyways, you can get an idea of how much variability there is in the model, how much uncertainty there is in the model selection. Some of the credible sets only include 4 models. Some include as many as, 22 models, right? Here's one that only includes 2 models. This happened to be a very long alignment. It's entire mitochondrial sequences.
So credibility has no notion about false negatives, false positives?
No. It's just probabilities of models.
So why don't you just list all and then you get 100% credibility?
You could, but then you're not learning something about how much variability there is. Okay. Yes, 203 models. So you can see that of those 200, we started with the uniform prior probability on the 203 models, and after observing this mammalian data set, it sort of reshaped itself, and most of the probability was on only 2 models. 95% of the probability was only on 2 models, and 5% was on the remaining 201 models. I'm not going to talk too much about this because I'm realizing I'm going to start running out of time, but basically there's other things you can ask. You can ask what different ways of choosing models, selecting models would do, and basically because people only look at a limited set of models, they test Chooks-Cantor versus Kimura versus GTR, you'd often stop in this procedure at intermediate values and would never actually even consider some of the models we were looking at because we expanded the candidate set of models to all the possible time-reversible models. And another thing you can ask is, well, what types of models were typically chosen? And usually they're models that don't constrain transitions and transversions to be equal to one another, which is sensible. People have often long realized that transitions and transversions seem to have different rates and sequences.
On that last slide?
Yes?
The first 2 models, is the second one sort of a superset of the first one?
Yeah, so this model here is nested within this model. Okay, now of course we also can do all this stuff that I've been talking about without requiring nesting of models, which was something you'd need for likelihood ratio tests, but not for Akaki information, for instance.
How sensitive were your models chosen prior?
I haven't explored that. Okay, and I can tell you that for amino acid data, but I think we should talk about that later. They can be sensitive, but not probably in this particular analysis. And then you could also ask, how did the named models? There's only like a half dozen of them, and here they are. That is models that have been described. And by the way, there's about 196 papers out there waiting to be written if you want to get your name in the literature 196 times, one for each model. But basically, the named models, if you looked at summed up the posterior probability of the named models, they generally had more posterior probability than they had prior probability. Bayes factor is greater than 1, which means that they're reasonable models, which is sort of what we expected. So here's some of the conclusions from this first part of the talk. Just that you can't actually treat these models as random variables in a phylogenetic analysis, and if you want then to turn the problem around, you can say that, well, we could also do phylogeny inference without conditioning on any particular substitution model. Okay, so I want to change track a little bit here and talk about natural selection in DNA sequences, and I'm going to be limiting my attention to protein coding DNA sequences where there's this idea that we can partition substitutions that occur on the tree into two flavors, one of which is non-synonymous substitutions. These are nucleotide changes at the DNA sequence level that change the amino acid. Or you can have a synonymous substitution, which is a change in the DNA sequence that because of the redundancy in the genetic code doesn't cause an amino acid replacement. And people often will summarize these types of, these estimates of these rates as a DN-DS rate ratio. And the reason why they're interested in this is that this DNDS rate ratio, which I'm going to call omega for historical reasons, has different interpretations in terms of what type of natural selection operated it's on it. So the usual pattern you see is one for most protein coding DNA sequences is a case where DNDS is less than 1. And the interpretation is this. If you're an individual that happens to have an amino acid replacement in a functional protein, chances are you're going to be screwing up the function of that protein, not improving it, right? Most changes that you can make, random changes you can make to a protein, aren't going to improve its function, right? And so such an individual will have a lower fitness and be removed from the, have fewer children, have a lower survivability, and such mutations would not be the ones you're going to find in the long term in the population. So that's the usual pattern you can see in protein coding DNA. Omega equals 1 is usually considered sort of neutral or neutral-ish type of evolution, considering that there's more going on than just amino acid replacements determining the fitness of an organism. Things like codon bias are known to occur as well. And then in some instances, you'll actually see DNDS greater than 1. And these are cases where individuals bearing an amino acid replacement in some protein have an enhanced fitness, right? And so you often see signatures of positive selection in viral proteins that are subject to immune surveillance by the host, for instance. A lot of reproductive proteins are in animals are known to be very rapidly evolving ones as well. And the framework I'm going to be looking at is these, basically, it's once again a continuous time Markov model, but now instead of modeling the substitution process on the nucleotide level where you have 4 states, A, C, G, and T, we're expanding the model around a codon, a triplet. Okay? So now instead of having 4 states, we have 4 to the third, or 64 possible states, all the possible codons. And so we're going to have a 64 by 64 rate matrix now, and this is how people often will parameterize such a matrix. Most of the entries of the rate matrix are zeros because they require 2 or 3 changes in an instant of time. So this is a 1 substitution at a time type model. But the others, you might have a transition transversion rate ratio, which would be kappa. And depending on whether the nucleotide change causes a replacement in the amino acid sequence or not, you might multiply the rate by factor omega for non-synonymous changes and not for synonymous changes. So basically, we now have a direct way of estimating this DNDS rate ratio. And this is work that goes back to the mid-90s by people like Zhiheng Yang and Nick Goldman and Spencer Muse and Brandon Gott. And this particular parameterization goes back to 1998. This is Nielsen and Yang, 1998. And so basic idea is you have some codon, let's say, and we're asking what's the rate of change to these other possibilities. And of course, there's a lot of other codons, but all those have rates of 0 because they involve 2 or 3 changes. Well, the first thing is one of these codons is a stop codon, and we removed the stop codons from the state space. So for the universal genetic code, there's 61 sense codons, so we have 61 states. But we can, basically we're partitioning things depending on whether the change is a transition versus a transversion or whether it's synonymous or non-synonymous. Now there's a couple interesting things you can do here, one of which is you can actually, so for a long time, I should say, in the early mid-90s, people basically assumed that you had a constant DNDS for all the sites. So basically, we did not allow DNDS to vary across a sequence, which is the biological that says we're not allowing constraints to change over a sequence. Okay? So starting in 1998, there was a couple interesting developments, but basically we have a model where the DNDS at a particular codon is treated as a random variable drawn from some probability distribution. And one of the simplest types of probability distributions you can think of is one where you have some number of categories. So here's a so-called M3 model from a later paper where you have 3 categories. Class 1 is you have 1 DNDS, or 3 different DNDSs with this constraint. And what you do when you calculate the likelihood for a particular column in your alignment, where a column now is a triplet of nucleotides, a codon, is you calculate the likelihood 3 times, once for each of these codons, and then you do the averaging. Basically, the average would be the probability of the data at that site conditional on it being the first category, conditional on the prior probability being the first category. It's just simple averaging. Right? And the important thing, what they did in these particular, or what people usually do is you have more parameters than just this DNDS. You have the tree, the branch lengths, the prior probabilities and so forth, the transition transversion rate ratios. What you do is get maximum likelihood estimates of all those parameters, and then you condition on them for further analyses. And one of the further analyses you might do is calculate the posterior probability that a site is in one of these 3 classes. Okay? And so this is a very common procedure now, I should say. So you can actually calculate the posterior probability that a particular site's in a 3rd class, for instance, category 3, just using Bayes' formula. And this approach where you condition on maximum likelihood estimates of all the parameters is called an empirical Bayes approach. so it's called an empirical Bayes. Basically, we're conditioning on all the parameter values taking maximal likelihood estimates or some good estimate. This is the approach taken in PAML, and you can take other approaches where all the parameters are treated as random variables. It doesn't matter. But the end result is that you can make plots like this, and you'll see such plots in a lot of molecular evolution papers, where the sequence is laid out along the x-axis, with this being the first site, that being the last site in the sequence. And along the y-axis, we have the probability that the site's in a particular category. In this particular paper, if you're interested in positive selection, you look at basically categories that are black. These are this is the only category of these three categories where the DNDS is greater than 1. So this sites in this class correspond to positively selected sites. And so you can see that this site has a high posterior probability of being in positive selection, as does this one, as does this one, and so forth across the sequence. Now the approach I told you about is cool because it relaxes the assumption that the non-synonymous synonymous rate's constant across the sequence, but what it doesn't do is it doesn't, once a site's in a particular class, it's in that class over the entire evolutionary history of the tree. And so I and some colleagues were interested in relaxing this assumption. And what we did is we took that model I just told you about, the 61 by 61 rate matrix, where you have three different classes, and we embedded those three 61 by 61 rate matrices. Each of these has a different DNDS value into a larger matrix, 183 by 183 rate matrix. So you can think of this new rate matrix as like a big tic-tac-toe board, where in this corner we have substitutions occurring according to one rate model. This substitutions occurring according to another DNDS value, and this substitutions occurring according to a third class. So when you see the code on AAA, for instance, AAA can be in one of three classes. It can be in class 1, class 2, or class 3. So what you have here along the state space is now the 183 codons, AAA1, AAA2, AAA3, and so forth. Along these other submatrices on the off diagonal, what you have is these represent switches from one class to another. So along the diagonal of these submatrices, you have switches. Along the off diagonals, you don't. So basically, this would be a switch from, for instance, AAA class 2 to AAA class 1. This type of model only allows a substitution or a switch from one class to another instant of time. It doesn't allow multiple events to occur. But basically, you can implement such a model. You change all the 61s in your code to 183s. And what we're going to do is we're going to take that empirical Bayes approach again. We're going to maximize the likelihood with respect to all the parameters. And the first thing we're going to do is I implemented such a model. And the first thing I'm going to do is look at how it performs on some real data sets. So I'm going to be looking at some serial samples of HIV from different patients that was published originally by Shankarappa et al. Basically, they sampled, had patients who recently were infected with HIV, came into the doctor's office, got the blood drawn, and from each blood draw, they would sample maybe a dozen sequences, viruses, and sample a particular gene. And they did this until basically every couple months until the patients died over about a 5 or 6 year period. So you have DNA sequences or alignments that vary in size. And so here I've got patient 1, 2, 3, and patient 4 was never published in that paper, 5, 6, 7, 8, and 9. And what I've shown you here is the likelihood. So one nice thing about the model I just described is it has a single parameter to the switching rate, which as it goes to 0, it collapses the models that people have been using traditionally, where you don't have switching. So this is the maximum likelihood estimate of the switching rate, and we're comparing it to a model that doesn't have switching. You can compare the log likelihoods, and you see the different parameter values. And so one thing you can do is you can do traditional likelihood ratio tests. So this isn't very Bayesian, of course, but I'm not that hardcore about things. So you can do likelihood ratio tests and ask, does adding that additional parameter improve the likelihood significantly? And across the board, it does. There's evidence, at least according to this model comparison, that selection is changing its action over time at individual sites. So, and you can also look at profile likelihoods for the switching rate. This gives you an idea of just how much uncertainty, and there's actually quite a bit in the parameter estimates for the switching rate. So what you see here is this line that you see drawn across is roughly 2 log likelihood units down from the peak. And basically there's asymptotic theory that allows you to construct rough confidence intervals from this. So you can say the confidence interval goes from here to here for this particular gene, where this line, the likelihood surface, intersects that line. So you can see in some of these cases the confidence interval is quite wide. And I'm using confidence interval like they use it. Here's a case where the confidence interval is narrow. Now, what that comparison does is allow you to make the blanket statement that it looks like this model is a better one. right? Adding that single parameter gives you a significant improvement to the likelihood, but it doesn't allow you to sort of scan through your data and pick out individual sites where selection's changed its action. And we can't use the formula I showed you earlier, this application of Bayes' rule, because now the process is a squirrely one, right? You can't count on it staying in the same class over the entire course of time. So what I end up doing is actually something very similar to what Vladimir talked about, but much simpler and much more naive than what he talked about, but correct still. What I'm imagining is that you have a branch which has a length of v, and just for the time being, we're assuming that we know both endpoints. We know it started in state 0 and ended in state 1, but we're interested in calculating what fraction of the time it spent in state 2. So this is the formula you would use. It's basically a transition probability from 0 to 2, which is here, over a branch of length s, and then from 2 to 1 over a branch of length v minus s, divided by the total probability of going from 0 to 1, which is also a sum of over the 3 different possibilities here. We just integrate that over all possible placements of that point, that dot on this branch. That gives you the dwell time, or the expected fraction of the time in state 2. And if we divide that by the branch length, you can interpret that as the probability that if we were just to randomly sample the branch at some point, we would have captured the process in state 2. And of course, what we have to do here is we also have to remove the conditioning on the beginning and ending states, but that's not difficult to do. What we're able to do then is we're able to, for these different patients, make graphs that look like this. Basically, this is patient 1. This is a tree where each tip is an individual viral sequence. And I've rooted the tree basically with, so in general, the sequences on the left are the ones that were sampled earliest in the infection, and the ones on the right were the ones that were sampled later in the infection. Most of the, and what we have, this is amino acid 10, this is amino acid position 52, and 132, 159. Most of the sites, most of the amino acid positions were all blue, meaning there's no evidence of positive selection at any time over the course of the infection. So I'm only showing you the interesting ones. So this particular amino acid has evidence of positive selection early in the infection, but not later, whereas these other 3 have evidence of purifying selection early in the infection, but positive selection later, right? And only affecting a subset of the sequences. And I thought it was kind of interesting that there's a lot of interest in co-receptor usage in HIV. Basically, usually when people are infected by HIV, it's the R5 version. They use the CD4 and the CCR5 co-receptor. And later in the game, there's often a switch in co-receptor usage to CRCX4. Those are called X4 variants. And that by eye at least, that change appears to be coincident. I'm going to skip that. So the main point here is that we can actually develop models that also allow selection to change over time. And in the last few minutes, I want to talk about one last thing. I'm not going to be able to talk about the very last thing I want to talk about. I actually have 2 more things I want to talk about, but I'm only going to talk about one. And this is going back and forgetting about changing selection over time, but once again, how we can model variability in DNDS across a sequence. Okay? This is going back to the original Nielsen and Yang paper in 1998, which is the first to allow or relax this assumption that DNDS is constant. And in a later paper, in a 2000 paper, Yang and Nielsen and Peterson and Goldman examined a large, instead of saying, well, let's, in addition to considering a model that had some number of discrete categories, they said, well, let's consider a model where the DNDS is a gamma-distributed random variable, or a mixture of gammas, or a beta on the interval 0, 1, or a mixture of a beta and a point mass between 0 and infinity, or a mixture of a beta and a gamma, or a mixture of betas and normals. You get the idea. They looked through every probability distribution you could think of. And they looked at discrete versions of these too. So a lot of these models start to look similar to one another in terms of how you calculate likelihoods. And so I was just wondering if there's other ways of accounting for variability across sequences, going back to this idea I talked about earlier on partitions. And so what I'm going to do is talk about a Dirichlet process prior model that allows DNDS to vary across a sequence, and I'll explain that in a second. But the general approach is we're going to get away from this empirical Bayesian approach, and everything's going to be a random variable with a prior probability distribution. And what we're going to try to do is base inferences of positive selection on what's called the marginal posterior probability distribution for DNDS at each site. And we're going to use the usual MCMC technology to approximate these things. So the thing that you may not be familiar with is the Dirichlet process prior. And it turns out actually a lot of the work on the Dirichlet process prior was done at Berkeley, which is interesting. Antoniak and Ferguson were both at Berkeley. Pittman picked it up. And now this fellow Michael Jordan, who's in stats and computer science, has done a lot of work on the Dirichlet process prior. It's called a non-parametric model for mixtures, right? So where the number of mixtures is an unknown variable. There's some connections to evolutionary biology and population genetics, and it's been used with limited by evolutionary biologists. And the way it was first explained to me and the way I'm going to explain it to you is in terms of the Chinese restaurant table process. And so this is the deal. So remember, it's a probability distribution on partitions. And so what we're going to imagine is we have data elements, in this case patrons, entering a restaurant with a countably Chinese restaurant. I don't know why, but it's always a Chinese restaurant with a countably infinite number of tables, and each table can hold an infinite number of people. So, but you're mathematicians, many of you. I guess that's not a problem. So they're going to enter one at a time, and the first person is going to sit somewhere with probability 1. All right, so now where's the second person sit? Well, the idea is that they can sit at the same table occupied by this person with probability 1, the number of people currently at that table divided by some normalizing constant, or they're going to sit at an unoccupied table with probability alpha divided by the normalizing constant. So alpha is this concentration parameter. It's one of the parameters of the Dirichlet process. Clearly, if alpha is big, you're going to be more likely to sit at unoccupied tables. So we let this person randomly enter, and he sits at that table. Now, this third person can sit at this table or this table with probabilities that are proportional to the number of people at each table or at an unoccupied table. Maybe they sit there. And this last person is going to be more likely to sit at this occupied table than this one because there's more people there. Okay? Or they can still sit at an unoccupied table, and so she sits somewhere else. Right? So you can see we've partitioned the elements among the tables. Right? And so it's a probability distribution on tables. And it turns out that you can calculate a lot of interesting probabilities, some of which I'll show you, and it doesn't matter the order in which these probabilities the order in which the patrons enter the restaurant doesn't matter. Okay? And just to complete things, you have to randomly get a meal. So here's mushu pork or something like that. So just to beat this to death, an alignment's going to walk into the restaurant. And so each site's going to walk in and sit at a table. Now the difference, what I'm going to be showing you is the sites don't walk in one at a time, but in triplets, right? The triplets, the codons are marching into the restaurant. But they're going to be marching into the restaurant, and the sites will be partitioned, and they don't eat things, they eat numbers, I guess. So each table is going to have written on it a parameter, and all the sites sitting at that table will share that parameter. Okay? And one of the nice things about and the parameter is drawn from some probability distribution, which in the literature is called G0 usually. So if you see it, it's usually called G0. And one of the nice things about this process, a lot of very smart people, Bayesian statisticians have been working on this problem. So a lot of the MCMC procedures have been worked out. Okay, there's some very good. So, this is theta. So remember, that alpha is sort of describing how clumpy the process is, and then there's parameters associated with the numbers on the table. So there's also things on the table that, you look down at the table and something's written on it. And whatever's written on it is the value, the parameter value you're going to be calculating your likelihood under. Okay. So there's two. I divided. I didn't use alpha here. I used theta because it's a different parameter. It's the probability. It's the number written on the table, and it's drawn from some probability distribution.
Does it make the table more attractive, or what does it do?
Well, so let's say this is the DNDS, so that we'll make this pretend it's omega. Well, if you're happen to be a codon, a triplet that has lots of non-synonymous changes, you would rather sit at tables that have a high DNDS. It's easier to explain. So here's some of the things you can calculate. There's usually this thing Z, allocation vector, which describes how the elements are partitioned. And K, the number of tables, you can calculate that. You can also calculate just the probability of the number of tables, the prior probability, the expected number, prior mean of the number of classes, number of tables that are occupied, or the probability, that two individuals are sitting at the same table. And this is just to remind you, they will. They don't here. This is just, I'm just looking at probabilities of the parameter values, of the partition, not the parameters that are assigned to the individuals. So that's being separated out right now. So in terms of what I'm doing here, it's mostly uniform prior on trees. Branch lengths have a particular prior. Codon frequencies have a flat Dirichlet distribution, not Dirichlet process, a different probability distribution. The DNDS rate ratio, the number that's written on a table, is going to be the ratio of two exponential random variables. So that's one of the things. Okay. And here's some of the data sets we looked at. Just protein coding DNA sequences, just to give you an idea of the number of tags, the number of sites. We're going to be doing the usual MCMC business in terms of approximating the joint probability distribution of everything. But to sort of cut to the chase, although we're integrating over everything else, this is the only thing we're really looking at. This is the marginal posterior probability distribution of the DNDS at, say, site 21 or site 56, 66. I'm not showing you everything clearly, right? And what I want to point out is, first of all, we ran these analyses with 3 different priors for alpha. Basically, we specified alpha such that the prior mean of the number of tables was close to 1 or 5 or 10. And this is just to show you that, that the prior on alpha can make a difference. So if you make alpha too small, then you can get these hump distributions, which basically you're forcing the data to be explained by very few tables. So a site's either sitting at one table it would not like to sit at, or another table it wouldn't like to sit at, right? But it's a compromise. Whereas if you have enough tables, then these marginal posterior probability distributions look very similar from one another. The other thing you'll notice is I shaded the fraction of the distribution that's greater than 1. And basically, the probability that a site's under the influence of positive selection is just a fraction of the time it's sitting at tables with DNDS greater than 1 in this case. And so that's what I'm showing you here is just a fraction of the time sites were sitting at tables with DNDS greater than 1. I'm laying the sequence out along the x-axis, and the y-axis is just showing the posterior probability of being under positive selection. Once again, each one of these panels shows if you use a prior mean of close to 1, a prior mean of 5, and a prior mean of 10 for the number of tables. And you can also pick out individual sites. Again, this is just showing you the sites ordered from the highest to the lowest posterior probability, conditioned on it having a posterior probability of greater than 0.95. And you can see that there's some Monte Carlo error perhaps in approximating these probabilities, but the ordering is more or less the same. So just to summarize this bit, I think that this Dirichlet process prior is a useful prior for this particular problem, but also for others in phylogenetics where you have partitions. And there's lots of other examples that you can sort of apply these types of ideas to. And this is, once again, a fully Bayesian approach, so all uncertainty in all the parameters is accounted for, in a sense. And I didn't talk about this too much, but the number of categories, number of tables is difficult to infer. But there seems to be information in a lot of data sets that the number isn't too small. All right, so I was going to talk about something really interesting, but I was overambitious. So if you want to talk, I can talk about later, if you're interested, we could talk about the uncertainty and alignment and genomic analysis, but I'm going to stop right here.
Questions?
Yes?
For small number of tables, is this useful? Or not?
So remember, the number of tables is a random variable. It's an unknown quantity in these analyses. So throughout the MCMC procedure, the number of tables is changing from one cycle to the next. But it's influenced by what type of prior distribution you put on that alpha, the concentration parameter. The number of tables is bounded with the smallest being number 1 and the largest being the number of data elements you have. Every element's set in a different table.
Any other questions? Okay, let's thank the speaker.